So this is our second lecture of general virology related with classification of viruses. First system is about Holmes 1948. This system of classification used linear binomial system. Linear binomial system to classify viruses. In this system, they used one order, that is virals, and classified all the viruses in this order viral. And further it was divided into three subgroups based on the host. If the host is a bacteria, or it is plant, or it is animal. So based on the host, this classification divided the viruses in three subgroups. So this is the phaginae, means these viruses attack bacteria, and for plant it was called phytophaginae, for animal it was called zoophaginae. So this was the Holmes classification, earlier classification system, and this system used Carolus Linnaeus binomial nomenclature. Binomial means genus and species concept to classify the viruses. They put all the viruses in one order, that is viral, and three subgroups or suborder that was based on the types of host. If it is host is bacteria, means phaginae means those viruses which attack bacteria. Phytophaginae means viruses which attack plants, and zoophaginae attack animal. So this system was further rejected by other authors. So after Holmes, another system was LHT system of viral classification. This system was given in 1962. It was based on physical and chemical characteristics. It was based on physical and chemical characteristics. These characteristics were mainly four characteristics were used in this system, and those four were NC, that is nucleic acid, whether it is DNA or RNA virus. C for capsid symmetry, whether it is helical, icosahedral, or it is complex. Number 3, naked or enveloped. Number 4, capsid diameter or number of capsomeres. So this was the LHT system. It was established in 1962, and this system was based on physical and chemical characteristics. And these characteristics were mainly four characteristics, NC. First is nucleic acid, DNA or RNA virus. Second C is capsid symmetry, then naked or enveloped, and last is capsid diameter or number of capsomere. So after this we'll go to next system of classification. Number 3 was ICNV, It was established in 1966, but further this name was changed to ICTV. In this ICTV classification, there are 15 hierarchical rank. There are 15 rank. They are realms, then sub-realm, then kingdom, sub-kingdom, then phylum, sub-phylum, order, suborder, then class and subclass, genus and species. 2, 4, 6, 8, 10. So realm, subrealm, kingdom, subkingdom, phylum, subphylum, order, suborder, then family. After this order, family, subfamily, then class. Mistake. So these are the 15 ranks according to the ICV taxonomy: realm, subrealm, kingdom, subkingdom, phylum, subphylum, class, subclass, order, suborder, family, subfamily, genus and species. Out of these 15, out of this box, these 5: order, family, subfamily, genus and species, these are 5 levels or 5 ICTV hierarchical levels. These are mainly used. After this, for example, if the species is FMD virus, even after species there are further classification for individual viruses. For example, in case of this FMD, there are pools, total 7 pools, then serotype, total 7 serotype, then topotypes, then lineages and strains. Topotypes means nucleotide difference is 15 to 20%. Lineages means nucleotide difference is 5 to 15%. And there are 3 strains which are used in the FMD vaccine. One is for O, one is Asia 1 and one is for A. So these are the strains, lineages, topotype, serotype and folds. These typings are further into individual viruses. But as per this ICV taxonomy classification, there are 15 hierarchical ranks. Out of these 15, 5 hierarchical levels are commonly used. So this was ICTV classification. Next is Baltimore classification of viruses. It was given in 1971. This classification is based on methods of or procedure of manners of mRNA synthesis. How the mRNA is synthesized. Which mRNA? This is viral mRNA. So mainly this method is based on mRNA synthesis. This classification further divided DNA viruses and RNA viruses. DNA viruses may be double-stranded, single-stranded. Similarly, RNA viruses may be double or single stranded. This is known as, in Baltimore there are 7 groups. So this is group 1, this is group 2, 3, and this is not 4. This single stranded is further divided into plus stranded, single stranded RNA plus sense minus sense. This is 4 and this is 5. So these are 5, 2 are left, and these 2 are this is RNA with DNA intermediate. RNA with DNA intermediate. This is group 6 and this one is DNA with RNA intermediate. This is group 7. So these are 7 groups. Classification by David Baltimore. These 2 groups are also known as single-stranded RNA reverse transcriptase group, and this 7th is also known as double-stranded DNA reverse transcriptase group. And one example for this is the retroviruses, and one example for this is the hypnoviruses. So this was all about the classification of viruses. Holmes classification, it uses Carolus-Linnaeus binomial system, LHT system based on NCNC. Then ICTV classification that use 15 rank. Out of 15 rank, 5 levels are most commonly used. Next is Baltimore. Mainly it is based on mRNA synthesis. Means in all these 7 groups, group 1, 2, 3, 4, 5, 6 and 7. In all these 7 groups, how the mRNA is synthesized. Based on this, there are 7 groups: DNA and RNA. Double-stranded, single-stranded. Group 1, 2, double-stranded, 3, single-stranded further plus sense, minus sense. So this system goes in like this direction. Group 1, 2, 3, 4 and 5. And opposite direction, there are 2 groups, number 6 and 7. Now we'll learn the classification for your entrance exam. Number 1, the normal structure of the DNA. As per the Watson-Crick, it is double-stranded. And the normal structure of the RNA, it is single-stranded. So we have normal DNA, double-stranded, normal RNA, single-stranded. These viruses, first we'll divide them into non-enveloped, without any envelope, and these are enveloped. These non-enveloped, these viruses are mostly they are circular, and these are mostly linear. There are 2 examples, A and P. This A is nothing but adeno and this P is papova. In this there are 2 virus classes, that is papilloma and polyoma. These are the double-stranded non-enveloped circular viruses. Enveloped viruses are, these are These are happy viruses. They are very happy because they have envelope. This H is herpes. Number 2, A is S for viridae. Number 3, this P is pox, and number E is happy, E is envelope. So these are A and P, these are happy viruses, and in this group there is one exception. And that exception is Hepadna. That is Hepadna virus. The full form of Hepadna, this H is helical symmetry. This H is helical. E for enveloped. PA means partial. D means double-stranded DNA, and one more is RT, reverse transcriptase enzyme is present. Because this Hepadna is exception, it comes into Baltimore group 7. So this is all about DNA viruses, double-stranded DNA. Then similarly, this single-stranded RNA will be divided into non-envelope, without any envelope, and envelope. So after this double-stranded DNA, we'll see the single-stranded RNA. Again, this RNA means single-stranded RNA. Just like DNA will divide into two, non-enveloped and this side enveloped. Non-enveloped means they do not have envelope. It means they have only capsid. Capsid means cap. These viruses, they have only cap. C-A-P. So these viruses which have only capsid or cap, they are cap viruses. This is Kelsey and Astro and Picorna. So non-enveloped means they have capsid, they have cap. So these are cap viruses. And these are the enveloped viruses. For these viruses, there is very simple trick to remember. A bunny born to crop fields. A baby rabbit, normally they born in the crop field. So this is the trick for the enveloped viruses. So here A means artery viridae and arena viridae. Bunny means bunia viridae. So this trick is about bunny born to crop field. Bunia viridae, born means this is Borna viridae. To means Toga viridae. Born to crop field. So we'll divide this crop into C-R-O-P. C means corona. R is, first R is retro, second is repto. Retroviridae. Then repto. O means orthomixo and P is paramix. So a bunny born to crop field. So last is the field. Field means Flaviviridae. From this field, one exception is here, that is Filoviridae. Philo. The viruses here, they are not infectious to our livestock. They are zoonotic diseases like Ebola, Marburg. These are two diseases. So these viruses are not infectious for livestock. They do not cause any disease in the livestock. So now to these viruses, as we know, RNA viruses also have plus and minus signs. How to remember plus and minus signs? All these, the number one rule is all non-enveloped RNA are plus. Means these are plus. All the three non-enveloped RNA, they are positive signs. They are plus. Then which are plus, which are minus? So very simple. The first and the last. First and last, they are plus. Flaviviridae and this artery, they are plus signs. Then next three are minus, next three are plus, next three are minus. So this is how you can easily remember. Second trick to remember is first and last, they are plus. First and last, they are plus. And then first three negative, then next three plus, then next three alternately negative. Because these Filoviridae do not cause any disease in livestock, so we call them negative signs. Because this is an exception to the rule. These are negative because they do not cause any disease in livestock. After this, we will see rule for symmetry. Rule for symmetry. The rule for symmetry means number one, all DNA, they have icosahedral. They have icosahedral except pox viruses. That is one MCQ for. Pox viruses have complex symmetry. So double stranded, single stranded, DNA viruses, RNA viruses, these are the normal one, and these two, these are the exception. So we have total 4 exceptions. These are the exception, that is single stranded DNA, and these two are also exception. 2 double stranded RNA viruses. So these non-enveloped are A and P, enveloped are happy viruses, and here they do not have envelope but they have capsids, so they are CAP, cap viruses. Here enveloped viruses are a bunny born to crop field, and in these exception 2 viruses are there, 2 are there. First is Sarcoviridae, that is circular. Second is Parvo, that is linear. And these are Birna and Rioviridae. So in all these viruses, first we'll see some basic rules. DNA viruses are mostly not plus or minus sense. So this plus minus sense, this goes to these RNA viruses. Which one is plus, which one is minus? There is one rule. So that rule is these non-enveloped, all non-enveloped RNA are positive sense viruses. They are positive sense viruses. And these four, 1, 2, 3, 4, all the four exceptions, they are non-enveloped. There is no envelope in these four exceptions. So our rule says that all non-enveloped RNA are plus sense, means these are plus sense, and these two are also non-enveloped, so they are also plus sense. So these five viruses, they are positive sense. For these viruses, the rule is first is plus, filled last is plus, then 3 times minus, 3 times plus, 3 times minus. This is how plus minus concept goes. And this Filoviridae, as they do not cause any disease in livestock, so we call them negative sense viruses. So these are mostly zoonotic like Ebola and Marburg. So that will keep for the exception portion. So plus minus is over, then symmetry. For symmetry, all DNA viruses, all means all DNA, they are icosahedral except pox. Means this adeno, yes, icosahedral, this is icosahedral, polyoma is icosahedral, herpes is also icosahedral, aster is icosahedral. The pox is exception. This is having complex symmetry. So this is about the symmetry for DNA viruses. So these DNA viruses are also icosahedral. For RNA viruses, the rule is all plus RNA are icosahedral. All plus RNA are icosahedral except corona, except the coronavirus. So we'll find all the positive sense, they are icosahedral. So this is plus, so this is icosahedral, plus means icosa, plus means icosahedral. Here this corona is the exception. This corona is it is helical. It is helical. Rest all plus. This one is also icosahedral. This plus is also icosahedral. This plus is also icosahedral, and this plus is also icosahedral. Remaining are the negative sense. So all negative RNA, they are helical. They are helical symmetry. So these negative sense all are helical symmetry viruses. Next is about the inclusion body. Next about the inclusion body. All, whether DNA or RNA inclusion body, they are acidophilic, also known as eosinophilic, except adeno and sarco. These two are basophilic. So this Cercoviridae, it is having basophilic, and this adenovirus, this is also having basophilic. Rest all the DNA or RNA, they have acidophilic or eosinophilic inclusion bodies. Normally, DNA viruses, they multiply in the nucleus. So DNA viruses produce intranuclear inclusions and RNA produce intracytoplasmic. But there are two exceptions. This ASFR and POX. These are DNA viruses, but they produce their inclusion body intracytoplasmic. So normally DNA virus intranuclear, RNA intracytoplasmic, with two exceptions. ASFR viridae, POX viridae, intracytoplasmic. And here the paramyxo, we know the canine distemper, they are intracellular and intranuclear both, but their color remains same, eosinophilic. They are eosinophilic because the rule is all DNA RNA are acidophilic or eosinophilic. Then rabies, rhabdoviridae, rabies also come in general rule, that is RNA means intracytoplasmic. So for the rabies, this is intracytoplasmic and eosinophilic. That is normal. So the last portion is the reverse transcriptase. There is only one virus from this RNA family which is having RT, and there is only one virus that is having RT. This RT is hepadnavirus, and here the RT is RT, that is retrovirus. They have reverse transcriptase enzyme, one from RNA side, one from DNA side. Hepadnavirus, and this is retrovirus. This one is Baltimore classification group 6, and this is Baltimore classification group 7. So this is all about the classification. DNA, RNA, their plus minus, their symmetry, and their inclusion bodies with exception.